Hello, and welcome to my poster presentation. My name is Anna Maria Kotsabasi, and I'm a master's student in bioinformatics in the University of Crete. This work is a part of my master thesis, which was conducted at the lab of Dr. Potamias at CBML in Forth, in collaboration with Professor Daskalakis from the Cyprus University of Technology. This presentation has the title Correlation between CYP2D6 variants and their metabolic activity by molecular dynamic simulations. As you may already know, CYP2D6 is one of the most significant enzymes in Cytochrome P450 enzyme superfamily that plays a fundamental role in the metabolism of many clinically important drugs, such as antidepressants, chemotherapeutics, beta blockers, and opioids. At present, more than 100 haplotypes of the drug metabolizing enzyme CYP2D6 have been reported in FarmVar, resulting in wide intra-individual variability in drug metabolism activity and changes of the drug plasma concentration. The complete connecting link between the genetic variants and the metabolizer phenotype is still an open and challenging question. Our main objective here was to investigate key factors that determine the metabolizer phenotype by appropriately employing molecular dynamics methods. With this approach, we aimed at providing information that connect the fluctuations and conformational changes of those variants to the crucial prediction of their function, as well as their effects on drug metabolism. This would be of great importance for areas like adverse drug reaction prediction, personalized medicine, and drug discovery. So we have chosen to probe the structural characteristics of several CYP2D6 star alleles with definitive level of evidence. And for this purpose, the first step was to select the crystal structure of CYP2D6, which we will use as initial coordinate to build the models. We selected the crystal structure 4XRY based on the completeness of the resolved CYP2D6 sequence, the resolution, and the quality of chain. Chain A, its corresponding heme factor and crystallographic orders were retained, whereas the inhibitor was removed. Afterwards, we appropriately protonated some of the residues in order to maintain the hydrogen bonding network within the crystal structure. We added the deep therapy water molecules, we added ions including potassium and chloride, and the all-atom models were embedded in orthorhombic boxes, and the charm protein force field was used for the residues and ions. For the production of atom classical molecular dynamics, the Newton's equations of motion are integrated. Classical molecular dynamic simulations of the fully solvated CYP2D6 variants with atom interactions described by the charm force field were performed in GROMACS. We ran 3 replicas of 1 microsecond for each allelic form, getting 30 trajectories in total, and we generated a series of configurations. After this step, we did the trajectory analysis and we tried to build a prediction model for the metabolizer phenotype. So I'm going to show you some interesting representative results of this work for 2 functional and 2 non-functional allelic forms. Root mean square fluctuations were calculated to evaluate the structural flexibilities. However, in order to draw a general conclusion about mobility of the different allelic forms, we visualized the structures by populating the B-factor column on the PDB files for the 10 different allelic forms. The color scheme indicates the degree of fluctuation, from blue indicating little fluctuation to red indicating large fluctuation. We observed that CYP2D6 variants with normal function remain in one more stable configuration, as shown here. In order to understand the extent to which the atomic fluctuation of a system are correlated, a dynamic residue cross-correlation analysis was performed using the Bio3D package as shown here. Here are the results for the functional variants and for the non-functional ones. In general, functional variants present more strong correlations between their residues compared to non-functional variants. The amount of correlated and especially the amount of anti-correlated motion for non-functional variants decreased significantly compared with that for functional variants. We investigated which correlation and anti-correlation relationships are present or absent in each case. Red and blue lines indicated correlated and anti-correlated motions respectively. In non-functional variants, anti-correlated motions were observed mostly between FF prime loop, LXF, BB prime loop, and KK prime loop. After completing the trajectory analysis, we continued our analysis with Markov state modeling for the CYP2D6 which is the wild type. Using data from our analysis and the Dika coefficients of specific residues, we built a prediction model of the metabolizer phenotype using the vanilla-like multi-layer perception. The results show that all allelic forms are correctly predicted to belong to their correct functional class. Here we can see the results, the prediction probabilities of 2 functional and 2 non-functional alleles. To sum up, in the present study, we have probed the dynamics of 10 different allelic forms of CYP2D6, functional and non-functional. We tried to find key factors that will help in the prediction of the metabolizer phenotype of this enzyme, and our results indicate that based on B factors, several sites of non-functional variants seem to be less stable in comparison with functional variants, and also the loss of correlation relationships in combination with the loss of anti-correlation relationships in case of non-functional variants can influence the functionality of CYP2D6. Both changes could be used as markers in the discrimination of the 2 classes of metabolizing activity. Also, we have developed a prediction model for the metabolizer phenotype, which could be of great importance for research in drug safety, drug repurposing, and drug discovery. At this point, I would like to mention that this work was supported by the computational time granted from the National Infrastructure for Research and Technology in the National HPC Facility, ARIS. Thank you very much for your attention.